This is going to be Chapter 10, Basic Human Physiology. And after completion of this chapter, you'll be able to understand cellular physiology and the need for homeostasis, fluid movement as dynamically driven by concentrations and hydrostatic pressure, the body's general adaptation syndrome, cellular-mediated sympathetic and parasympathetic responses, the body's thermoregulator mechanism, and the cell's ability to adapt to variable conditions. So, first thing we want to talk about here is physiology is a study of the body's function in its normal human condition. And the processes of this can be both physical, mechanical, and biochemical. Cellular milieu. Human body is a complex association of independent cells which form tissues. To survive, cells need an environment, and that is the milieu. Water, food, things like glucose, amino acids, and fatty acids, and oxygen. All elements must be maintained in an internal environment where there is sufficient heat and acidity for biochemical reactions, or metabolism to occur, and the life processes to go on. Homeostasis. One of the most important and the primary function of the human body is to maintain a relatively even state of temperature, acid load, oxygenation, blood glucose, and so on. Everything kind of needs to be in balance. Wellness is a state of physiological equilibrium, and homeostasis is the body's process to attain internal equilibrium. Homeostasis is always battling against external forces like cold, heat, lack of food, infectious diseases, and so on, and internal conditions as well, like diseases. The body must overcome these to maintain homeostasis, and these factors are always changing. So the body in this process is in a continuous state of flux. It adapts to the external and internal conditions while trying to maintain a normal environment or range. The sum of all the biological processes within the body, circulation, ventilation, and everything else, through interaction with one another, support the cells of the body and help maintain homeostasis. Cellular physiology. We have some terms here that we're going to go over. Cell wall membrane is the outside of the cell. It's porous, it's semi-permeable. It's a dual layer lipid protein matrix. Cytoplasm. That would be the internal fluid that all the organelles float in, primarily water and organelles, subunits with specific cellular functions and things like nucleus and things like that we're going to talk about here. Next term is the nucleus, which is the center of the cell. It's the first organelle to develop. Contains chromosomes and DNA, the blueprint for cellular production and reproduction. And then we have lysosomes. They're outside of the nucleus. So we got a cell and a nucleus, and we're talking about lysosomes. It would be in this part here. Lysosomes are inside the cytoplasm, tiny sacs that contain enzymes which break down proteins. They break down foreign proteins and bacteria as well. Examples of this would be antigens, or the cell's own chromosomes as well, if the cell's starting to destroy itself or its lifespan is ended. Lysosomes help kind of pin it up. Mitochondria, the largest organelle in the cell. Inside the mitochondria, glucose is transformed into an energy source, adenosine triphosphate, and the mitochondria is inside of here, and it kind of looks like a kidney bean, if you will. Used to power the rest of the cell's functions. Nickname is the powerhouse of the cell. The body must maintain a complex set of conditions to actually work properly, and having enough energy is one of those. And these are all the things that we just got through talking about. We have the nucleus. Let me see if I can find. There's a lot more stuff in here than we were just talking about. The lysosomes help break down the cells. This right here, that all this stuff's floating into, is the cytoplasm. This would be the mitochondria. This is a phospholipid bilayer, the cell membrane. So be sure you're familiar with these. Sodium-potassium pump. A cell pretty much needs energy. Needs energy in the form of adenosine triphosphate, to be more specific. Anaerobic metabolism is the first thing that the chapter talks about here. Quick overview. Cell changes the glucose into pyruvic acid, which is in turn converted into lactic acid by an enzyme, which is lactate dehydrogenase. And this process only yields 2 ATP molecules, and that's about 2% of what is achieved in aerobic metabolism. Now, aerobic metabolism is with oxygen. Kind of a peculiar note in the same area here on my notes that I'm looking at. The blood cells or erythrocytes that carry oxygen cannot use the oxygen that they carry. They depend on anaerobic or without oxygen metabolism and save the oxygen for the cells of the body instead. The other term that's in this section is aerobic metabolism. And a quick overview of it. Body uses 8 different enzymes to divide glucose by a process of glycolysis, breaking down or dividing sugar, to create pyruvate. Now, pyruvate and oxygen enter into the citric acid cycle or the Krebs cycle. And complex series of things occur in the mitochondria, and ATP is created. Now, we create a whole lot more ATP, 36 molecules, than we do in just the breakdown of sugar, which was 2. The end result creates carbon dioxide, water, and energy in the form of ATP and heat. Quick point up here before we leave this page. The sodium-potassium pump requires energy. It is how your muscle cells generate an action potential or actually contract. Glucose storage. When abundant, glucose not needed immediately is stored in the liver and the muscles in the form of glycogen, and glycogen is a polysaccharide. When glucose levels fall, the body retrieves glucose from these stores. Glycogen is broken down by an enzyme called glucagon. And glucagon is a hormone found in the pancreas. Alpha cells secrete this in the islands of Langerhans. If the body requires glucose, and there is none for storage, it can create glucose from other substances, and this is called gluconeogenesis, or the production of new sugar. The process of aerobic metabolism is dependent on many organ systems working together in synchronization in order to be effective. Fluid balance. The body is primarily made up of water, 50 to 55% of the total body weight in women, and 55 to 60% of the total body weight in men. Water is intracellular and extracellular. The majority of the body as water is actually intracellular, which is about 35 to 40% of the total body water is intracellular. In the extracellular fluids, we have 2 categories, interstitial fluid and intervascular fluid. In the interstitial fluid, this is about 16% of the total body water. And in the intervascular fluid, this is going to be about 4% of the total body water. Now, an anatomist refers to spaces, and this would be intercellular, extracellular, those kind of spaces. There's only 3 spaces as far as an anatomist is concerned. The first space is fluid inside the cells, and that would be intracellular. The second space is fluid in the actual bloodstream, and that would be in the actual intervascular space. And the third space is the volume of blood in between cells. And that's why we say sometimes if people have edema or something or swelling, or they move fluid in between the cells, like in ascites inside the belly or inside the abdominal cavity, we consider this to be a third space fluid shift. Nutritional flow, also in this section. Constant movement of fluid between compartments. Fluids can move from outside to inside the cells and from compartment to compartment by diffusion and osmosis. And this is a breakdown. This is figure 10.3 in your book. Distribution of water in the body. We have intracellular fluid, interstitial fluid, intervascular fluid, which is this little blue area here, and then cerebral spinal fluid. out of these four things that we just talked about, intracellular fluid is up here in the yellow, and extracellular fluid, everything outside the cell. Now this is broken down into interstitial, intervascular, what was in the blood, and then cerebral spinal fluid. The effects of isotonic, hypotonic, and hypertonic fluids across the semipermeable membrane, and this is 10.4 in your book. This depends on the tonicity of something, or how thick something is. So let me see if I can get something drawn here really quick so I'll explain this a little better. If we had a semipermeable membrane and lots of parts over here, this was thick, if you will, with all of this solute, and we only had a little bit of solute over here, and this side B, if you will, and side A. Side A was a solution, but it was a really thick solution that had lots of solute parts to it. And then a side B solution, which was really thin. This had a lot of water to it. Well, if we had permeability to go both directions, the solutes would diffuse and try to balance this like a teeter-totter, if you will, on both sides until all parts were equal on side A and B. The water, however, would move in this direction, and I changed ink color so you could see. Now, this is the definition of diffusion and osmosis. Diffusion, going from an area of higher concentration to an area of lower concentration. And osmosis, which is the diffusion of water, going from an area of higher concentration, there's more water over on side B, to an area of lesser concentration, which there's less water on side A. So essentially, osmosis is moving this way, and diffusion is moving this way. The tonicity of these fluids matter because this is what we put into the body, and from that we get net fluid movement. So a hypotonic solution, let's start up with this one here, and this is our semipermeable membrane again. Again, side A and B. If we introduced a hypotonic solution into side A, the hypotonic solution does not have near as many solute molecules as side B does. So we are going to get a net movement of water into the intracellular compartment. And an example of this, all of these compartments work off of these principles. So if you're giving somebody a slightly hypotonic solution, an example of this would be dextrose 5% water, and you're giving this to them intravenously, this will have a slight osmotic push into the intracellular compartment. And the reason being is a hypotonic solution is a little thinner than the solution that's in the body, or the tonicity of the body. So this is going to go inside the cell. Next one we're going to talk about is going to be isotonic. And an isotonic solution is equal on both sides. So we shouldn't have any net movement on both A and B. And this would be things like normal saline, lactated ringers. Hypotonic solution, slightly hypotonic, is dextrose 5% and water. And then hypertonic solutions. Let's talk about it. Now, a hypertonic solution, water flows out, and we're talking about these things right here are the cells. So in this one here, in the isotonic, equal movement. In the hypotonic, it has a net movement inside the cell because the liquid that we're putting in is thinner. And then in a hypertonic solution, let's talk about that one here really quick. Again, back to the semipermeable membrane, sides A and B. This has, now side B has more water on it. And side A, we're going to introduce a really hypertonic solution. So lots of solute parts. Now what's going to happen is osmosis is going to cause fluid movement in this direction. The reason being is there's a higher concentration of water on side B than there is on side A. Again, going from an area of higher concentration to an area of lower concentration. Important terms and concepts. Hydrostatic pressure. Hydrostatic pressure is created by the force behind the volume of water. An example of this, there's some terms in here that we're going to talk about. Another one is edema, which is a dramatic fluid buildup in the actual tissue or swelling. We've seen probably edema by this point. Isotonic solution is a balanced solution, and we talked about that in the last slide. A hypotonic solution, the fluid has more water and less salt or electrolytes than the solution on the other side of the semipermeable membrane, or has less water and more salt than the solution. So either way, what happens is the hypotonic solution is thinner, so it's going to go into the tissue. If you have an example of this, water is the most hypotonic solution that I can think of. And if you introduce it into the intravenous, just plain water, it will burst the cells because it will never balance that equation. It will rush in there until it destroys the phospholipid bilayer or the semipermeable membrane. Oncotic pressure, the force similar to that of osmotic pressure created by proteins. And if we think about it, as osmotic pressure kind of as the pull, that works a little bit better and it's easy for us to kind of remember that. Osmosis is going to be the diffusion of water, which is going to go from an area of higher concentration to an area of lower concentration. So in osmosis, we're going to get that. In oncotic pressure, the oncotic pressure is going to pull because this area here is thicker, or it has more solute to it than this area here. So the oncotic pressure, or the colloidal osmotic pressure, is going to have a considerable amount of pull to it. Albumin, the most common intravascular protein, principally made in the liver, third spacing. Whenever colloidal osmotic pressure or oncotic pull is low, fluid leaks from the intravascular space and into the interstitial space. So we get a third space fluid shift, if you will. And this is figure 10.5, the effects on oncotic and hydrostatic pressure upon the intravascular volume. The hydrostatic pressure is actually blood pressure. It's what pressurizes the system. So, again, one more time. Semipermeable membrane, and this is the cellular environment. This is side A and B. Let's start with that. So, if we introduced a solution on side A, let's back up one, sorry. If we introduce this solution on side A, side A's osmosis or osmotic pressure to push into side B, that would mean that there was more water on side A than side B. The oncotic pull to the same movement because of the thickness on side B, this is the oncotic pull. So it is pulling from side A to side B. And then hydrostatic pressure, this whole system will not work without pressurization. And this would be, in the form of the body we're looking at, this would be blood pressure. Temperature regulation. Enzymes operate best at about 98.6 degrees, plus or minus 1 degree. We have hyperthermia, which is too much heat buildup, and we have hypothermia, which is too little heat. Other important concepts, the anterior hypothalamus regulates body temperature. Vasodilation, one of the most effective heat dissipating mechanisms that the body has, vasodilates, and its evaporation off of the surface of the skin, controlled by the parasympathetic nervous system. Surface capillaries under the skin react to dissipate heat, causes skin to act as a massive radiator, and allows heat to dissipate by conduction, convection, and radiation. If the body should start to cool, then the hypothalamus can either increase heat production by causing shivering, or an involuntary contraction of the muscles and or vasoconstriction. Now, temperature and the oxygen hemoglobin dissociation curve. I put this in there. I went out on the internet and I kind of found this. This is an oxygen hemoglobin dissociation curve. And if we'll notice that normal condition is the purple line here, which is the line in the middle. I'm going to kind of go down it here just a little bit. And then I would like you all to notice this. So an increase in hydrogen ion, which would be more acidic, an increase in the amount of CO2, or increased temperature will cause what is considered a right shift to this curve, which essentially will make it more efficient for the body to offload oxygen. So fever is actually beneficial to the healing process at many levels. The hypothalamus controls mean body temperature during an infection. Poisons and bacteria stimulate chemical mediators to the affected hypothalamus. Reset the hypothalamus thermostat and cause the temperature to rise. These pyrogens, or fever producers, therefore create a pyrexic or fever environment, which is kind of hostile to the bacteria. It also moves the oxygen hemoglobin dissociation curve to the right, improving oxygen offloading to the cells. Metabolism at the site also increases. So fever is kind of a good thing with some control to it. If you have a fever of 106, 109, that is not a good thing. So low-grade fevers, 100, 101 is kind of a good thing. It increases metabolism and gets the bacteria out a little faster. Stress in the cellular response. Stress is constantly changing conditions caused by the body to respond and efforts to regain homeostasis. And Hans Seil pretty much studied rats for a little while, and when studying these rats, the rats' physiological response that they had to these external stressors, which were injections, he made them cold, injured them as well. And from this, he came up with something called the general adaptation syndrome stages. And there are 3 of them. There is an alarm stage, a resistance stage, and an exhaustion stage. So I got a picture of this on the next slide, but in the alarm stage, the body responds to stressors. In the resistance stage, the body attempts to regain homeostasis. In the exhaustion stage, or recovery stage, exhaustion occurs and the body's response is insufficient to meet the challenge of the stressor. There are 2 types of stress. There is distress, stress that overcomes the body's innate defenses, and there's estress, which is daily stress and essentially keeps the body's defenses on guard for larger stress threats. So this is kind of a good stress that keeps us moving forward. Other things that we talk about in this section, stress in the autonomic nervous system. The autonomic nervous system is automatic control over things that we have no thought process over. There are 2 divisions of it. There is the parasympathetic and the sympathetic. The sympathetic nervous system is pretty much the accelerator, and the parasympathetic nervous systems are the brakes on things. They have nicknames. Sympathetic nervous system is nicknamed fight or flight, because whenever we get into a stressful situation or we get those type of chemical receptors going, we get agitated and excited. We're ready to either run, flight, or go toe-to-toe with something and fight them. And then the parasympathetic nervous system has a nickname called feed or breed, and it kind of applies the brakes, if you will. If we're digesting a steak, if we're doing sexual things, this nervous system will take the front on it, so to speak. They communicate with one another through something called neurotransmitters. and these are chemical messengers. Neurons do not physically contact an organ that they innervate. The neurons are separated by a gap called synapses. Nervous signaling is transmitted across the synapses by neurotransmitters to awaiting chemical receptors across the synapses called neuroreceptors. Adrenergic transmission of the nervous signal using adrenaline, and this would typically be from the sympathetic nervous system. The parasympathetic nervous system uses another neurotransmitter called acetylcholine. Cholinergic transmission of the nervous system signal uses acetylcholine as a neurotransmitter at the motor end plate. The neuroreceptors, the parasympathetic nervous system has two of them. They are nicotinic and muscarinic. And the sympathetic nervous system has actually four of them. They have an alpha-1, alpha-2, a beta-1, and a beta-2 receptor. Stress and the endocrine system is also talked about here. The two effects of stress. Initially, the sympathetic nervous system stimulates the medulla and the adrenal glands and the endocrine glands to secrete the hormone adrenaline. Circulating adrenaline then goes to the liver and muscles where it stimulates glycolysis, or the breakdown of sugar, of glucagon stores, and that would be turning a polysaccharide into individual sugar molecules, and liberate glucose into the bloodstream. Simultaneously, sympathetic nervous system stimulates the release of corticotropin-releasing hormone from the hypothalamus. Now, what this does is this releases things like vasopressin and cortisol, which is a glucocorticoid, or your body's own steroid, and this can actually suppress the immune system. So it also brings up in the same section the immune system. Stress and the immune system, major organs are directly innervated by the nerve fibers of the autonomic nervous system. Hypothalamus acts through the pituitary gland and then the adrenal glands to modulate the immune response. Important function of the immune system may be to alert other organs of the impending systematic threat. This is done through the release of inflammatory mediators and interferons. So, immune system getting involved in this and being prepared for inflammation, if you will. So these are the general adaptation syndrome stages: the alarm stage, resistance stage, and then exhaustion stage. Please look over them. This is figure 10.6 in your book. And this is the sympathetic nervous system. Here at figure 10.7. And then at figure 10.8, neuron transmitter across this neuron transmission across the synapses. This is where it would release, down here, neurotransmitter between the synaptic cleft, if you will, across the synapses. This is the parasympathetic nervous system innervations. And if we'll notice, this is pretty similar to the sympathetic. For every yin, there's a yang, if you will. For every sympathetic innervation, there's a parasympathetic break. Cellular adaptation. Atrophy. The verbiage on atrophy is reduction of cells. Physiological loss of cells as a result of normal changes, so atrophy. Pathological loss of cells, if it's atrophy from pathology, is a result of a disease. Next term we're going to talk about is hypertrophy. Hypertrophy is an increase in either the weight or functional capacity of a tissue or organ beyond its normal. And an example of this would be a bodybuilder who undergoes resistance training to create and enlarge muscles, if you will. Cellular cell replacement, metaplasia, comes to term. Replacement of one adult cell type with another type of adult cell. Hyperplasia, an abnormal increase in the number of cells due to frequent cell division, reproduction, which is caused by tissue or organ, the organ itself, to increase in size. This may be physiologic, compensatory, or pathologic whenever we get hyperplasia or metaplasia. Dysplasia, too many new or immature cells that are non-functional, is what dysplastic or dysplasia means. Apoptosis, which is a process of planned cell death. So cellular death is apoptosis. And the process may be a pathological condition, It could be caused from an intrinsic or an inside or an outside source. To prevent intrinsic biochemical errors from accumulation within the cell, or to simply replace old, worn out, or old cells, all cells are programmed to essentially commit suicide, and this would be apoptosis. Maintains a relatively even number of cells among rapidly dividing cells. It also removes damaged cells, this process. So, in conclusion, review of the conditions necessary to maintain cellular viability or wellness, and thus human life, shows that physiology is a very complex process. The body's failure to maintain that internal environment or milieu through a dynamic ongoing process called homeostasis would lead to illness and disease. References for this lecture are from the Professional Paramedic Volume 1, Foundations of Paramedic Care, First Edition, pages 156 through 173, Delmar Learning. And if you have any questions concerning this chapter, please feel free to give me a contact or give me a holler. My name is Roy Smith. I can be reached at smithr@imsa.net or 405-219-7613. Thank you.